Now we talk about different NGS platforms. Remember, next generation sequencing is abbreviated as NGS, so we will use this term a lot. There are different platforms, NGS platforms, which are there, and four of them are the most famous one. Number one is 454, that is a subsidiary of Roche, and then there is Illumina, then there is Applied Biosystems, and then there is Torrent. 454 was founded in 2000 by J.M. Rothberg. Since 2007 and up till 2016 it will be a property of Roche, and but Roche is not supporting 454 any longer after 2016. It is located in Brantford, Connecticut, and if you guys want to see where Connecticut is, so that is the east coast of USA. You see that red dot over there on the US map. Their famous machines are GS Junior system and GS FLX Plus system. We can compare different sequencers using these 5 different parameters like read size, throughput, reads per run, accuracy, and runtime. So read size is, as you guys know, that when the sequencers sequence the DNA, that is into the small fragments. So we will call those fragments as reads. So here, if we compare these reads, GS Junior produces 400 base pairs, longer reads as compared to FLX Plus, so that produces 700 base pairs. If we compare the throughput, throughput is actually how much data is processed into a machine. So remember, there is an input to the system, and then there is throughput or processing within that system, and obviously finally we get the output. So if we compare the throughput, how much data is processed within the machine, so 35 MB for GS Junior, and FLX Plus can process 700 MBs. It's a million base pairs. If we compare the reads, how many reads are produced? They are 1 million in both of them. 100,000 for GS Junior, whereas GS FLX Plus produces 1 million. As per their claims, both the machines are accurate up to 99% or even more than that. So if we compare the run times, FLX takes almost twice the time of GS Junior. So Junior takes 10 hours and FLX Plus takes 23 hours. Illumina, which is the public property, it is registered with NASDAQ, which is the American Stock Exchange. It was founded in 1998. So far we can say it is the most commonly used or successful sequencing platform because of their sequencing strategy. It has the revenues which were recorded in on the last quarter of the last year. So till 31st December, its revenue was 512 million. Illumina is located in California, which is in the west coast of USA. Here are their famous machines: HiSeq, HiScan SQ, Genome Analyzer, and MySeq. Comparing these 4 machines with the same properties, we see that MySeq produces the longer reads, where we have 250 base pairs, as compared to Genome Analyzer 150 and 100 each for HiSeq and HiScan. Throughput is 600 GB for HiSeq, so it is good to study the different important biological questions where we have lot of read data. For example, the expression studies. MySeq produces 6 GB, small amount, so maybe it's good for targeted sequencing. I will talk about these things in a later section. As far as the reads per run, HiSeq gives 3 billion reads. If we compare MySeq, that is the least one, 1.2 million, and Genome Analyzer gives 320 million, and HiScan gives 750 million reads. Accuracy, again, over 99% as per their claims. Same way, based upon the throughput and based upon the production, they are taking the similar amount of the days. So HiSeq takes like 11 days, whereas Genome Analyzer takes the maximum 14 days, and MySeq is the minimum that takes like 1 day to 1.5 days. Applied Biosystems Incorporation, commonly known as ABI, again a public property, founded in 1981 in California, and it merged with Invitrogen, and then it became Life Technologies, which was later on purchased by Thermo Fisher company. The website is down there if you guys are interested in to see. Same way, located in the same city where Illumina is, same state in California. So here are the famous different sequencers as far as this ABI machines are concerned. So we have 5500 XL solid sequencer. We also have 5500 XL and a simple 5500. If we compare their performances, read size is obviously same for these three which are compared here, 5500 XL, 5500 and 5500 W. If we look into the throughput, obviously W is processing 360 GB of the data. GB here is giga base pairs, so giga is actually 10 power 9, or we can say 1000 million. Reads per run again for 5500 is the maximum, 6 million. Accuracy as per their claim is over 99%. So all of them are taking 1 to 2 weeks for completing a sequencing run. Life Technologies also have iron machines, which are kind of technologies. We can place them between the second and or next generation sequencing and the third generation sequencing. So they are kind of in between these two. So they are also located in California. Their famous machines are Proton and PGM, personal genome machine. So if we compare, read sizes are the same, 200 base pairs for both of them. Throughput is 1 GB for PGM, and Proton is processing huge amount of data, around it may be anywhere between 10 to 100 GBs. Same way reads produced per run, 1 million approximately to 660 million in Proton. Runtime is similar, so kind of quicker and faster machines. So far what we have seen that there are 4 important NGS platforms, which are 454, ABI and Illumina, as well as OinTorrent.